Ya puedes iniciar, Doc.
Buenas tardes para todos. Muchas gracias por su asistencia. Espero que les haya gustado el video que acabamos de compartir sobre nuestra Universidad de La Salle. Y pues mi nombre es Liliana Chacón. Yo soy la directora del doctorado y la maestría en agrociencias. Y bienvenidos a nuestro webinar. Nuestro doctorado y nuestra maestría en agrociencias contamos con 5 líneas de investigación que en la maestría las llamamos de énfasis. En estas 5 líneas de investigación hemos estado realizando en este año pues varios webinares. Cada uno de nuestras líneas está apoyando estos webinares. Quisiera recordarles nuestras 5 líneas son gestión y biotecnologías de la reproducción, aplicadas al mejoramiento genético en animales entrópicos, medicina, poblaciones y salud pública, agricultura sustentable, estudios agrarios y ambientales, y en el día de hoy estamos haciendo un webinar que corresponde a sistemas de nutrición y alimentación animal sostenible. Pues me complace presentarles a la doctora Carolina Vespayoc, quien es la coordinadora de esta línea, y muy amablemente hoy nos ha organizado un webinar, y pues la dejo a la doctora Carolina para que les presente a nuestra invitada especial. Doctora Carolina, a todos muchas gracias, y se me olvidaba comentarles, ustedes tienen en la parte de abajo a la derecha la opción para participar en el chat. Por favor, cuando seleccionen allí, van a escribir, seleccionen a todos los panelistas. Ahí tienen la posibilidad de seleccionar, entonces ustedes le dan ahí, escriben para todos los panelistas, y de esta manera podemos ver lo que ustedes se están preguntando. Cualquier duda, allí estamos atentos en el chat. Doctora Carolina, doctora Fernanda, muchas gracias.
Muchas gracias, Liliana. Bueno, buenas tardes a todos. Es un gusto tenerlos acá en esta tarde. Muchas gracias por atender nuestra invitación. Hoy este webinar tiene con el objetivo de traer ahí una tarde de discusiones sobre este tema que Fernanda viene trabajando, y creo que va a ser una tarde muy, muy interesante. Antes que yo presente la doctora Fernanda, la presentación del seminario se va a hacer en inglés. Entonces, Fernanda va a intentar como hablarlo despacio. A cada bloque de sesión ella va a hablar de 3 grandes temas. Entonces, al final de cada grande tema, ella va a hacer un resumen en inglés, y ahí después yo hago una intervención en español, porque la idea es que como para que todos puedan seguir acompañando como la presentación sin ningún inconveniente. Pero si ustedes tengan algún inconveniente a que esté hablando muy rápido, o que como que necesitan alguna otra intervención más en español, solo escriban ahí en el chat. Voy a estar muy atenta para que todos aprovechen bien esta tarde de discusión.
Como pueden
Entonces,
notar, el acento de la doctora Carolina es brasilera, habla portugués. Entonces aquí tenemos 3 idiomas: español, inglés, portugués. La doctora Carolina ya lleva varios años en nuestra universidad, pues, tiene ya un muy buen español. Entonces, Fernanda pues también habla portugués, inglés, y ojalá aprenda español también. Por ahora queremos también pues motivarlos para que usemos esta segunda y tercera lenguas. Entonces, por eso hemos querido que la charla esté en inglés. Sin embargo, pues si ustedes tienen alguna duda, nos van escribiendo. Muchas gracias. Chao.
Bueno, entonces voy a presentar nuestra súper invitada del día, Fernanda. Ella es zootecnista, graduada por la Universidad Federal de Pelotas en Brasil. Hizo su maestría en la Universidad de Oregon, del Departamento de Animal Science, sobre la orientación del doctor Máximo Bionas. Hizo su doctorado en la Universidad de South Dakota sobre la orientación del doctor Johan Osorio, y actualmente está desarrollando un postdoctorado en la Facultad de Medicina en la Universidad de Arkansas. Entonces, Fernanda tiene bastante experiencia en los temas de inmunología, biología molecular, y fisiología. Entonces, y el tema que ella va a presentar hoy es sobre Fecal microbiota transplantation. Entonces, Fernanda, el micrófono es suyo. Muchísimas gracias. Al final te agradezco de nuevo, pero ya al inicio, muchas gracias por su disponibilidad, su tiempo de estar acá compartiendo su conocimiento con nosotros. Yo voy a apagar mi video para que esté mejor la conexión, pero si necesita de algo, pueden escribir al chat o me dice por acá, estoy muy atenta. Also, your microphone is off.
I think it's working.
Yeah, now we can hear it.
Okay. Hola, buenas tardes a todos. Muchas gracias, doctora Carolina, y muchas gracias por la invitación a todos de la Universidad de La Salle. And that's all my Spanish. I'm so sorry. I wish I could know more to actually be able to give a talk in Spanish, but I'm not there yet. As Carolina mentioned, I did all my graduate school working in the animal science, biological science, and now I am a postdoctoral fellow in actually the human science field, and that's why I'm so excited to be here talking to you all today, because I like this comparison or this comparative type of approach. I have a background in animal science, and I can also do research of human subjects. And that's actually my passion, to be able to combine and do the translational work or translational research from one side, from one field to the other field. Okay, so to start today, our talk, here is a very brief outline of what I will show you today. As Carolina mentioned, I have a take-home message, so we can have like some stops between sections. I'll be talking basically three big subjects, and if you have any questions, or if you think I'm talking too fast, or you cannot understand me well, please just say it. I can try to rearrange. So overall, I will try to work with you through the factors that affect the neonatal gut microbiota colonization. So we know that there are prenatal factors during birth and after birth, that they impact in different ways the gut microbiota colonization of both neonatal calves and as well as babies. And with some data that I will show you from my own research and research from others scientists, we are going to try to look how this gut microbiota colonization can affect the overall inflammatory response and health of both babies and neonatal calves. And at the end, I will show you two experimental or let's say clinical trials, one in neonatal calves and one in babies, that we use something called fecal microbiota transplantation, that I will be always refer to it as FMT. I always like to make very clear that I'm not a clinician, I'm not a physician or pediatrician. Everything that I will show you today is based on literature search, is based on the articles that are published, they were peer-reviewed, and they are based on either in vitro trials or clinical trials in both animals and humans, and I have no further conflict of interest. So to start our talk today about understanding all how FMT can be used and why it can be used and how the gut microbiota colonization happens, we need to understand this first slide here. I know it's heavy, looks busy, but we can walk through every single factor that is written in here. So the infant gut microbiota can be affected by different factors, as I just mentioned. Here we have a combination of prenatal factors, meaning factors during the pregnancy of the mother that will affect the gut of the baby. I would like to start pointing out the placenta here. As far as science goes, this is a question mark. Some scientists have shown some evidence that actually the placenta can have its own microbiota, and therefore the fetus in the uterus would not be completely sterile, as we thought that was the case for many, many years. However, some doctors and other scientists, they do not believe that is true. There are some other evidence showing that actually all the studies that have shown that there are some bacteria in the placenta, and if that can be transmitted to the fetus, is actually contamination during birth or at the collection of the placenta tissue. So the fact of how the placenta can actually affect gut microbiota or microbial colonization, that is still a question mark. But there are some other factors that they are very well known, and they are very well characterized, that they impact the gut microbiota colonization. One of them is maternal diet, for example. We know that the maternal status, maternal health status of the mother can affect since birth, during birth, the gut microbiota of the babies. For example, obese mothers or mothers that they are overweight, we know that the gut microbiota profile of those women, they are slightly different than the microbiota of a healthy mother. And that have been shown in clinical studies when those babies grew up, when they are 2 to 3 years old, that the obesity that happens during infancy can be related with that maternal healthy status of the mother. And the tendency is that we call something like transmission of obese microbes, that actually the profile that we see in obese mothers, we end up seeing sometimes in those children as well. And that's why we have here metabolic related disorders. Another factor that is very well known and very well characterized that can affect the baby is the maternal infection. So any type of infection that the mother has during pregnancy will affect the microbiota colonization, the first bacteria that will colonize the babies. Another factor that is very well known is the antibiotic treatment, and I think that's also very well known worldwide, that any antibiotic, even in adults, even in our case, if we receive antibiotic treatment, we know that at least during a short term, during a short amount of time, our microbiota composition will change because of the antibiotic treatment. And that is true when it comes to the gut colonization of babies as well. Other factors that I think we are more used to hear about it is the mode of delivery. We know that babies that they are vaginally born versus babies that they are born from C-section, they have a different microbiota composition during the first 2 weeks of life. And we also know that term versus preterm babies, meaning term babies represent the babies that they are born after above 37 weeks. And preterm is any delivery before 37 weeks. And we know that these, the babies, the preterm versus the term-full babies, they also show a different microbiota profile during the first weeks of life. One factor that I will talk more about today is the breast feeding versus the infant formula feeding. That's a very well-known factor to impact in a different way and have different health outcomes in babies. The solid food, the introduction of solid food that clearly change the gut microbiota profile of the infants. And lately, we have seen that environmental factors, even pets, babies that have exposure to pets when they are 6 months, 1 year, 2 years old, versus babies that don't have pets, families that they do not have pets at home, there is also some speculation how much that can impact. And of course, you probably hear about it, that probiotics, not just the supplementation in formula feeding, but also probiotics that the mother could have during pregnancy will affect that gut colonization as well. And all of these factors, they do not stop influencing during just the neonatal time or neonatal period. We know that there are some correlations or associations with respiratory diseases and allergies, and as I mentioned before, obesity or other metabolic related disorders when those babies, they turn into kids or children. And it's very well known that the shift from how much these factors impact the gut to when we start seeing an adult-like microbiota is around 3 years of age. So as I mentioned a lot about the mother, here is just an example of how much the maternal microbiota or the different mother microbiomes can affect that baby. So here we have the example of the different profiles of microbiota coming from the mother. There is a classic profile in the oral cavity of the mothers. The skin, that's why we always, let's say there is a scientific reason behind of more than the affection between the mother and the baby right after birth. That skin contact is it's transmitting microbiota as well, and a healthy microbiota in this case. We know that the stool microbiota of the mother is reflecting the gut microbiota composition of those mothers, as well as there is a vaginal, very well characterized vaginal microbiota. And that's why vaginal delivery is so important when it comes to gut microbiota colonization. So, excuse me. So here we have just another way to show you how the infant gut microbiota is impacted. So there are two, let's say, terms that you guys have to keep in mind. That is very, we use a lot when we talk about microbiota composition. One is the alpha diversity. That is a simple measure of the species diversity, or the different number of species that we see from each baby or from each sample. And there is another term that is the beta diversity. This one is the variation of microbial communities from different samples or different environments. And why this is important is because it's very well characterized that in neonates, the alpha diversity starts as low right after birth, as in the photo here of newborn, and then start slowly increasing with age, meaning when they are newborns, their alpha diversity is low because they are not exposed to many factors, right? And until 6 months of age, we see predominant species, predominant bacteria, that they are very well associated with two factors, delivery mode and the neonatal diet, meaning if the babies are breastfed versus formula fed. And then after 6 months of age up to 2 to 3 years is when we start seeing an increase in the alpha diversity. Because the babies after 6 months, probably they will start in solid food, exposure to more subjects and more factors, and that increase the number of species that we see. And with the beta diversity is the opposite. We have a higher beta diversity of microbiota at the beginning of life, and then we start seeing that it decreases with age. Here is another example about how the infant gut microbiota colonization happens. I know it's another very busy graph or slide, but it's very important to understand how this happened, especially up to 2-3 years of age, because these are all associated with all those perinatal factors that I talked at the beginning. So here we have the newborn around 6 months to 1 year of age, and then 2 to 3 years of age. So at the beginning, we do see some gram-negative bacteria, especially a lot of times we see that in the C-section babies. And that is just as part of the normal intestinal flora, just for the fact of birth itself. Then we see this family here, they are gram-positive bacteria. And this family, they are very well characterized in babies up to 6 months of age, because they are directly associated with the milk, either breastfeeding or infant formula. And I will show you in few slides ahead that this family here, they are called bifidobacteria, and they are very high abundant in the breastfed babies. And there is a reason for that, because there is something that I'll talk in a little bit. There is a bioactive component in the human milk. So when you breastfeed the baby, you see this bifidobacteria very high during the first weeks of life. Then we have the bacteroides, that is, they are normal components of the intestinal flora. They are high abundant through life, including in adults. And then we have this one here that has these famous bacteria that they are the firmicutes. And these ones, when the gut microbiota establishment finishes around 3 years of age, and in the adult gut microbiota, these are the predominant bacteria that we see. And then we have Clostridium, that is very well known, not just for the pathogenic ones that cause infection, but also the ones with nutrient digestibility. And these ones here, that they are anaerobic bacteria, and they increase in the gut of humans through time. And here we have another way of viewing this infant gut microbiota colonization. As I mentioned before, the alpha diversity, it's lower in children or in babies. And here on the right, we have the abundant microorganisms in adults. And what this plot means is that here in children, you can see that there are less colors, right? We don't see so many different colors when we compare with the bars here that represents the adult gut microbiota. So here we have a lower alpha diversity of microbiota, and here we can see the increase in the adults. So basically, this one here is bifidobacteria, as I just mentioned, the predominant one when it comes to breastfeeding. And then we see bacteroids, that they are also highly abundant in babies that have infant formula and through all life. And then in the adults, we start seeing more, let's say, types of microbiota composition. Now, through all those factors that I mentioned at the beginning, of course, I will not talk in detail or in deep through all of them, but I select two of them here, the delivery mode and the breastfeeding, that I believe they are the predominant factors during the first weeks of life in babies or in humans. And they are the factors that have been very well characterized, that all the clinical trials that we have, they point out to these specific factors. So here on the left, we have the delivery mode. As I mentioned, the difference in the microbiota profile between the vaginal delivery and the C-section born babies. So here we can see clearly that the vaginal the vaginal delivery babies, they have high abundance of Bacteroides and bifidobacteria. And when we look the C-section babies, they lack on those microbiota. And we start seeing more Clostridium-related microbes. Now, when we look to the plot in the right here, that's about how breastfeeding impact, we see that the babies that they were breastfed, they have high abundance of bifidobacteria. That is a very important bacteria for the infants, that I will explain a little bit why. And in the ones that they are formula fed or infant formula fed, we see the Bacteroides being the predominant one. And there are, we need to understand that there are so many factors that they can impact the gut colonization, and depending on the region, depending on, for example, here in the US, some factors might play more of a role compared to the babies in Colombia, just based on the geography, on the environment as itself. But delivery mode and breastfeeding, I will emphasize again that they are the most important, crucial factors, at least during the first weeks of life. Here is just an example of why human milk or breast milk or breastfeeding, it's so detrimental for the gut microbiota colonization. So we know that human milk is a biological fluid, right? There are so many components in the human milk. For example, we do have commensal or beneficial microbiota present in the breast milk or human milk. We have different hormones, we have different antibodies such as IgG, IgA, that they are essential for the passive immunity in neonates. We have also here, I hope you can see, these are the famous human milk oligosaccharides, or HMO. They are complex carbohydrates that they are not digested by the infant's small intestine, but they reach the large intestine of babies, and there where they play a role not just in the immune system, but also in the production of short-chain fatty acids. And here they are. And we know that these human milk oligosaccharides play a role. They are a crucial component of the breast milk. Not just because all these factors that we know HMOs have an impact, have an effect in the infants, but is about the fact of how much of these HMOs are present in milk. HMOs, they are the third most abundant solid component of the human milk. And that's why we emphasize a lot about breastfeeding and how breastfeeding is important, because these human milk oligosaccharides, they are unique to breast milk. We cannot find these HMOs in infant formula unless you feed the babies with a supplemented infant formula that can have some synthetic HMOs. So after all of this that I show you today, I hope was not confusing, because there are so many factors involved. But the take-home message here is that we have several factors before birth, at birth, and after birth that will impact the gut microbiota composition. We will see in the future slides that all of the microbiota profile during infancy can affect the inflammatory response as well. And here, briefly summarizing, between birth up to 6 months of age, when we have exclusively milk feeding, we see bifidobacterium, bacteroides, and lactobacillus as the predominant microbiota profile of the babies. And then when they are towards the establishment of that gut microbiota, we see a microbiota profile that looks more like our microbiota profile, our adult microbiota composition that is composed of firmicutes and bacteroids. Carolina, do you want to go?
Sí. Entonces, resumiendo acá lo que nos dijo Fernanda, entonces hay factores que van a afectar esta diversidad de la microbiota intestinal desde la gestación, el parto y luego después del parto. Entonces ella hizo énfasis ahí en todo lo que es principalmente el modo del parto, si es un parto natural o una cesárea, y también el tipo de alimentación, si es una alimentación con leche materna o con leche de fórmula. Entonces que esto va a afectar directamente la diversidad y la población de esta microbiota intestinal. Entonces y los estudios apuntan que el establecimiento de esta microflora intestinal está muy relacionado con factores inflamatorios y trastornos metabólicos que pueden se presentar en la vida adulta. Entonces, que los bebés ahí en los primeros 6 meses de vida, cuando son alimentados exclusivamente con leche, entonces presentan principalmente estas 3 clases de bacterias ahí presentadas en la diapositiva, y próximo a los 2-3 años de edad, ya presentan una microflora mucho más diversa, mucho más semejante a la que presentamos nosotros los adultos.
Gracias. So, keep going with our talk. Now we are going to switch from gut microbiota colonization in babies or humans to neonatal calves. But before I start talking about the neonatal calves specifically, I'd like to leave you with a couple of questions to think about, it, like food for thought. So if all these factors that I show you, we know they affect the babies, and we know that the mother-baby connection also involves gut microbiota transmission. Would that be true for the cow and the calf as well? Or how much the cow can, the factors or the gestation or the infections that happen with that cow, how much can impact the calf? We know there are some evidence, there are some studies that they show some of that impact, but I think that we have so much room to look, to investigate, so other factors that we don't talk about that much. And there are a lot of room for extension work between researchers, scientists, and the extensionists, and do that translational work to the actually farmers. One thing that I start thinking about since I start doing this type of comparative work between humans and animals is if mothers can transmit microbiota through the skin contact between the mother and the baby, how is that transmission of microbiota or oral microbiota from the cow to the calf right after calving, when the cow is trying to lick the baby calf? So that's one thing to think about it, and we can discuss later after the whole presentation. And other thing that I start thinking also, and I would like that you guys think, it's about the colostrum management, right? We know that breastfeeding is so important, so crucial for babies. What about feeding beyond the colostrum time? Most of the farmers, at least in the US, they just feed colostrum on the first or second feeding after birth. And after that, they automatically transition to pasteurized milk or milk replacer. And what I would like you to think to do some type of extension work, what if we increase that transitional milk? Instead of feeding calves with pasteurized milk or milk replacer starting at 2-3 days old, what if we start that after 1 week? What if we keep the mother's milk that we know it's richer during that first week of lactation? So those are all questions that I don't have all the answers, of course, but I like for us to think about it. So with that being said, here is some factors very similar with the human infants, how factors can impact the neonatal calf gut colonization. So we know that some of the maternal factors are also true. They can also impact the microbiota diversity in the calves. As I just said, the colostrum, for many other reasons that we know, it's crucial for the survival, actually, of the calf, but it can also impact the gut microbiota colonization. As I just said, for you guys to think about it, the difference in the gut microbiota profile that pasteurized milk versus milk replacer can have in the diversity composition of the neonatal calves. One factor that the discussion is new, we don't have that much evidence to talk about it, but might be some microbiota profiles differences when the calves are housed in group versus individual pens. At least here in US, most of the farms, we keep the calves in individual, we call hutches or little houses. Another factor that is well known in animals too is the antibiotic treatment, that at least at short term, it completely changes the gut microbiota composition. And just like in babies, the introduction of the solid food shift the gut microbiota composition, as well as the feeding mode. In this case, for the calves, we would be looking at bucket versus bottle fed. Here is just an example to show you all how the prenatal factors and the factors at birth, they also impact the animals, neonates. So here we have some first colonizers that they were identified in the meconium, that are the first poop from any subject. So as early as 6 hours after birth, these special lactobacillus that is high abundant in the vagina microbiota was also identified in high abundance in the fecal samples of those calves. And what I was saying about the alpha diversity in humans seems to be somehow similar in the neonatal calves as well. Now, when we look at the first month of age in neonatal calves, the gut microbiota profile, when compared to babies, they are a little bit different. So here, the plot on the left, we are seeing the relative abundance of the main bacterial phyla, identifying the feces of calves from 1 day old up to 30 days of age. And also we have here the representation of two different farms, farm 1 and farm 2. And on top of that, we are also seeing the difference in the microbiota profile between healthy calves and calves with diarrhea. So the plot in the left here, it's we don't see that much difference. We can see that overall the calves at 1 month of age, they are showing high abundance of firmicutes. That is the predominant bacteria in the adult gut. And then we see some of the proteobacteria, and here in green, the actinobacteria represents the bifidobacteria. So maybe the bifidobacteria that is so important from the breastfeeding factor in babies might be playing a role in the neonatal calf gut colonization as well. But one thing that it's different is the microbial diversity that can be impacted by disease and environment, at least based on this study. Here on the right, we are seeing a very simple index to measure bacterial diversity, and they were able to identify some differences between healthy and calves with diarrhea when it comes to the diversity of the microbiota. And also, if you look, these two here, they are representing one farm, and these other two, another farm. So the environmental conditions of each farm, in this case, also plays a role in that gut microbiota colonization. Here is another evidence that was just published this year, and I think it's very interesting because it clearly shows when which age those calves they have a very different microbiota composition, and with which age they start being very similar. So, for example, this red here is the gut microbiota composition at birth, right after birth. And then here we start seeing week 1 of age, 2 and 3. And somehow here, during the first weeks of life, we do see that the microbiota profile is diverse. However, when we look here, when those calves are older, between 6 to 8 weeks of age, that very likely they have been weaned. They had already started eating solid food, compared to these first 3 weeks here that they were exclusively on milk feeding. You can see that they are more similar in that age, meaning that they are transitioning to a more established gut microbiota composition. And why that is, why it's so important to understand how the gut microbiota colonization is impacted. We know that there are, just like babies, there are different infections that they happen during the neonatal stage. I won't talk about it, how the immune system develop after birth, but it's well characterized that a lot of neonates, including calves, they are immune suppressed during the first weeks of life because the immune system will start developing or maturate with age. And that's another reason why we see a high incidence of diarrhea when those calves are between 2 to 3 weeks of age. And it's very important to keep in mind, because now there are some evidence showing that the microbiota profile can be related or associated with health, versus calves with diarrhea. And here is one of the evidence that was recently published. It's a study just published this year, if I'm not wrong, like last April. And here you can clearly see that some of the first colonizers, such as bacteroides and firmicutes, they are stable. There is no significant changes between healthy and calves with diarrhea. However, here we see a difference. So calves that had diarrhea, they had an increase in the abundance of these two bacteria organisms. And in a little bit, this one, especially this one here, will make more sense for you all. But the take-home message from all these plots is that the microbiota profile of the calves can prompt calves to have diarrhea. And if this is the evidence, some factors might contribute to the increase of this specific bacteria. And what we could do is to find a way to intervene and avoid the increase of this specific bacteria. And the take-home message here from the gut colonization in neonatal calves is that we have very similar factors impacting the calves as we have seen in babies. Some of them, they are not very well characterized as in humans, but all the environmental factors and especially the feeding type during the first weeks of life will impact that neonatal calf. And that's even more important in calves because we know these calves around up to 3 weeks of age, they have a suppression in their immune system. So, and as the evidence that I just showed, there are some specific bacteria that prone calves to develop diarrhea. So with that in mind, we need to find a way to intervene, or when is the best time, with what age we should do some sort of modulation of the gut microbiota composition to avoid infections, avoid diarrhea, and so on. And I will start talking with you about FMT in few seconds.
Ok, então, resumiendo acá lo que nos dijo Fernanda, que los factores que afectan el desarrollo de la microbiota en los terneros son muy semejantes a los que afectan a los bebés humanos. Y pensando especialmente en el punto de que estos terneros ahí alrededor de la 3ª semana de edad fisiológicamente presentan un estado de inmunosupresión. El objetivo es, y la alta ocurrencia de diarreas en estos animales. Entonces el objetivo de las investigaciones que viene desarrollando Fernanda y que ella nos va a contar enseguida es buscar estrategias para modular, o sea, para que este momento de inmunosupresión, de esta diarrea, pueda afectar negativamente el desarrollo de este animal.
Gracias. So I have to speed a little bit. So again, these are my disclaimers and very important. I will be talking about fecal microbiota transplantation since we have feces very well available. So do not try that at home. And at least I don't know how it's the regulation in Colombia, but at least in US, we cannot do any procedure, at least in humans, using FMT if it's not pre-approved by FDA. So FMT is the transplantation or the transplant of feces from a healthy donor after a heavy screening. So a very healthy fecal composition of a donor directly into the gut of any human or animal. At least in the US, the FMT has been used a lot and it's approved as a treatment just for this disease here, this infection. and upon federal approval can be used to treat other diseases. But as of right now, FMT is an investigational treatment. But the evidence that we have using FMT to treat this type of infection here are very exciting, and that's why more research start being done using FMT in babies and in calves. One of the reasons is because FMT can restore the healthy microbiota. So upon this disease here, for example, we know that there is a decrease in the commensal or in the good bacteria in the gut. And after FMT, this procedure can restore that good bacteria in the gut. And here is one example that I'll be talking about is how FMT was performed in babies. And this study was conducted in Finland. So they compare the vaginal born babies with the C-section born babies. And then they select some babies that were born through C-section, but that the mothers wanted to participate in the research, and so they fed like through tubing directly in the gut of those babies a fecal sample from the mother. So here is how they did. They mix 100 milligrams of healthy feces from the mother of those babies with the mother's own milk, and they fed that to those C-section babies within 2 hours after birth. And what did they observe? They observed very positive results in the case that the C-section babies that received the FMT mix in the mother's own milk, they somehow had similar gut microbiota composition with the babies that were vaginally born, and that's a very positive effect. So here we have the diversity plots, and you can clearly see just looking into the plot. The vaginal babies are the blue line here, and then the C-section babies that did not receive anything are the ones in red that you can see in both plots. They are down here. And the black line, they are the C-section plus FMT babies. And then you can see that in both cases, the gut microbiota diversity of the C-section babies that got the FMT looks like the vaginal born babies. Now here, the same study, they went deep to see which species or microorganisms were being impacted. And again, we observe the same thing. A very positive effect, the babies that were born through C-section and that they receive FMT. This procedure, the FMT, was able to restore the gut microbiota of those babies to look like the gut microbiota of the vaginal born babies. That is a healthier microbiota when compared to just the C-section born babies. So here we have the impact of this procedure of FMT on the delivery mode, and both together they can impact in a positive way the gut microbiota composition. Now, we actually, during my PhD, we were able to do the same, but in calves. So how we did that FMT? So in this case, we select not the mothers of the calves, but we randomly select the healthiest cow of the herd, of the farm, and we use that cow as the donor feces. And we mix in the milk replacer of those calves those feces, and then we fed them for 1 week, for a couple of days. And then we evaluate all the microbiota composition and overall health. So we did the experiment from birth until 7 weeks of age. As I told you at the beginning, here in the US, that's how we raise most of the calves. They are in individual houses. And all the animals, all the calves that we use in the experiment, they were fed antibiotic-free milk replacer, so we don't have any potential confounding effect here. And they were all weaned at 7 weeks of age. The solid food that we fed those calves were also antibiotic-free. And we mix, as I mentioned before, the feces in the milk replacer of those calves. And here is how we actually did it. From the donor cow, we mixed 25 grams of feces in the milk replacer, and then we bottle fed those calves with like a chocolate milk from 8 days to 12 days of age. And here is what we observe in those calves. Very interesting results when you think that we are feeding feces to those neonatal calves. We did observe a positive effect in their overall growth, both in body weight and Witter's height. If you're not familiar with Witter's height, here is a picture of how we measure that as an overall growth measurement for the dairy calves. And now, before I show you what we observe in the blood of those calves, we collect blood every week since 24 hours after birth until 3 weeks of age, and we measure the inflammatory biomarkers as well as the immune cells. And I know I'm hurrying, but before I show you the blood results, I need you to understand how these blood biomarkers of inflammation happen. So here you have a photo of a very beautiful enterocyte or intestinal epithelial of the gut. And here it's what happens when we have an infection in those animals. We see an increase in in all inflammatory receptors, pro-inflammatory cytokines and transcription factors. And those, when we have an infection, all of these pro-inflammatory cytokines will be released in blood. And then from the blood, we can have these pro-inflammatory cytokines. They can do the connection or signal the liver that there is an inflammation going on in the body. So the liver will do something to help with that inflammatory response that is called acute phase response. And we will increase the production of these proteins here, and we will decrease the production of these proteins here. But what we need to keep in mind is that these two proteins here, they're supposed to be producing the liver in a healthy state. And here we have our results of the FMT in those calves. On the left here, we have the haptoglobin, that we can clearly see that 2 weeks of age after the FMT. So here this black arrow is showing when we start feeding the feces, mixing the feces in the milk of those calves. So we observe at 2 weeks of age a significant difference in the haptoglobin. It's an inflammatory marker. But at 3 weeks of age, when we finish the FMT, we see that that concentration was similar to the control. So FMT induced a short-term inflammatory response, but also was able to end, not turning to a systemic inflammation. And the same was observed for the interleukin-1, that is another pro-inflammatory cytokine. After we performed the FMT, at 2 weeks of age, we observed a significant difference in the blood of the calves that receive FMT. And the same pattern with a positive health outcome at 3 weeks of age was actually lower than the control. So take-home message, FMT induced a short-term inflammatory response, which is a positive thing, but was able to not turn that into a systemic inflammation as well. And here we have the negative acute phase proteins that I mentioned in the previous slide, that these two proteins, they supposed to be produced by the liver in a normal state when the animals are healthy. And here we observe that at 3 weeks of age, the calves that had the FMT, the albumin was similar to the control, which is a positive effect, meaning the FMT did not induce a systemic inflammation, or at least did not impact the liver function of those calves. And the same goes for the paraoxonase, this other protein here, that we could clearly see at 3 weeks of age, higher concentration in the blood of the FMT calves. And this here, I don't have much time now, but it's so exciting because it's brand new data. You guys are seeing that I'm showing this for the very first time. It's not published yet, but we have here the microbiota composition of the donor. Mainly the green here are the firmicutes, and the blue here are the bacteroids. So the donor microbiota composition of the cow really looks like the adult human gut microbiota. And here are the diversity index. The 3 plots here, they represent the same the same index of diversity, but in a different way. But using the 3 of these diversity index, we observe a significant difference in the microbiota profile of the calves. So here in gray, we can see the diversity of the cow microbiota. And here at week 1, 2, and 5, in black are the control, and in white are the FMT calves. And we can clearly see that before the FMT, comparing to week 5 here, after the FMT, we had an increase in the gut microbiota diversity in the FMT calves, which could be associated with the donor microbiota composition as well. We won't go into deep into the details here, but I just want to point out that firmicutes and bacteroids were the predominant phylas, or the predominant microbiota observed in the calves, all of them, control and the calves with FMT, and they were also the predominant in the donor, in the cow microbiota composition. However, it was very interesting that we observed this group of bacteria here, that actually after FMT at 5 weeks of age in the calves that receive FMT, that was significantly lower. And I'm saying that it's very interesting because it's the same group of bacteria that cause diarrhea in calves from another study. So that's a positive effect from the FMT in the gut microbiota profile in our study. So take-home message, that's my last slide. You guys should be happy about it. So here we have the take-home message here is that FMT can impact the gut microbiota colonization of infants, as well as of dairy calves at least. And this procedure called FMT can induce at least short-term changes with both the research in infants and our in calves. We did not measure long-term effects, but at least as a short-term treatment or intervention, it can shift the gut microbiota profile, as well as we observe some positive effects when it comes to the inflammatory response. And with that, I would like to acknowledge and thanks this is my PhD supervisor. All the FMT in calves that I show you, we performed that experiment at South Dakota State University, funded by NIFA. And I would like to thank you all for your attention. I'm sorry that took me longer than it was supposed to be. And Universidad de La Salle, and especially Dr. Carolina Giacometto, thanks for having me. And I'm free if we have any questions, would like to further discuss anything.
Fernanda, muchísimas gracias. Solo como haciendo un resumen entonces de la última parte que nos dije Fernanda sobre el trasplante de esta microbiota fetal. Entonces que los resultados tanto en los niños como lo que se ve en los terneros no son estudios a largo plazo, o sea, no se conoce el efecto de esto a largo plazo, pero sí los efectos a corto plazo son positivos en el cambio de esta microbiota intestinal. Entonces, mostrando un buen potencial de aplicación para tanto para humanos como para los animales de producción. Bueno, Fernanda, yo sé que acá no tenemos mucho tiempo, pero si alguien tiene alguna pregunta específica, pueden enviarnos ahí en el chat, ponga a todos los panelistas y aquí yo con mucho gusto paso a Fernanda. Pero quería que comentaras con nosotros, ahí si quieres en inglés o en portuñol o cómo te sientas más. Todos estos estudios como utilizando todas esas herramientas genómicas, moleculares, súper interesantes, pero pensando en cómo transmitir esta información para el campo, o sea, en la práctica, ¿qué podríamos hacer o si está haciendo algo de esto, cómo lo podríamos aplicar?
Yeah, I don't think I can explain that in Spanish. So, yeah, that's a very good point. That's why we do this type of research at the end. We need a translational, right, to the farmers, to the extension people. The FMT, what I like in at least in animal production is that it's a cheap treatment, right? You just need to be able to screen the feces of a donor cow or a donor adult animal, and you can use that. We don't have, at least in the US, that's what I'm saying. I have no idea how is the regulation in Colombia, but at least here for animals, we don't need federal approval, and that's a positive thing. I do think that using that as, of course, first you have to screen the feces of your donor cow. You have to know that there is no pathogenic bacteria in that fecal sample. But instead of using antibiotics, we can come to a point that we can use feces. We can use fecal microbial transplantation to shift that gut microbiota instead of using antibiotics, for example, or in a way, as I said before, some sort of inflammatory response is a positive thing. We cannot allow that to turn into a chronic inflammation or acute inflammation. So using FMT to prone or to challenge the immune of the calves during those first weeks of life can be a positive thing. We are not there yet in that evaluation, but I believe with the right fecal sample, the right screening, we can get into a point that we can do that.
Y por acá nos comenta la doctora Liliana Chacón, como pensando en los diferentes tipos de sistemas productivos. Entonces, porque ahí usted nos estaba hablando de la creación individual, la cría individual de terneros, de bovinos lecheros, ¿cierto? Pero pensando esto en otros sistemas como el doble propósito, que es muy común acá en Colombia, o los sistemas, mismos los sistemas lecheros, donde se crían los terneros de forma colectiva. Esto, ¿cómo se podríamos aplicar este tipo de investigación o si los resultados podrían ser diferentes con este tipo de manejo en los animales?
Yeah, that's another good question. Thinking about FMT as a procedure, I'm not sure how it would work when we have a group of calves housed in the same environment, in the same place. For sure, we can expect a difference when it comes to the gut microbiota composition if you compare two individual hutches to the calves housed in individually, like we have here. But what I like to think about the group pens when you have many calves in the same housing, it's how that interaction, because I'm sure that the gut microbiota profile of the calves housing group versus the individual will be different. I don't know how different. I don't think we have the evidence showing the clearly difference. But for sure, as I mentioned, the environment will play a role. And we don't know if that's a positive or negative, right? We know for behavior or diarrhea, if it's positive or negative, but when it comes to the microbiota composition, would be very interesting actually to compare group housing versus individual housing and how different they will be. I don't know how different they will be, but I'm sure we will have a different profile.
Sí. Don Fernanda, pues para encerrar esta nuestra tarde de discusión, te agradezco nuevamente en nombre de la maestría y del doctorado en agrociencias. Ya la doctora Liliana, si quiere, se despide también, pero personalmente para mí es un orgullo muy grande verte, que yo te conocí en los primeros pasos en la investigación y verte presentando estos datos de tanta calidad y toda esta carrera tan fantástica que está empezando. Y agradezco su disponibilidad en estar acá compartiendo toda esta información con nosotros y espero que en un futuro próximo te tengamos acá personalmente y podemos desarrollar investigaciones en colaboración.
Gracias a vos. Y sí, quiero ir a Colombia.
Gracias, Fernanda. Eso me parece muy bien y ojalá podamos seguir apoyando. Y nosotros tenemos, pues, en la línea que coordina Carolina de nutrición, creo que allí podremos apoyar mucho a nuestros estudiantes. Tenemos en este momento varios estudiantes que están trabajando precisamente en internet y en producción lechera. Yo creo que allí podríamos, pues, recibir muchos aportes de tu parte, Fernanda. Y muchas gracias por la compañía, por la excelente presentación. Y qué bueno que la juventud nos supere. Claro que Carolina
Gracias a
está muy joven.
ustedes.
No, no, pues muchas gracias por todo.
Gracias a ustedes por having me.
Y a todos los participantes también. Muchas gracias. Cualquier duda, recuerden que en la página de la Universidad de La Salle pueden encontrar allí los programas académicos. Está el doctorado y la maestría en agrociencias, está toda nuestra información, así como los correos míos y de cada uno de los coordinadores. Donde dice más información, pueden bajarlo y allí está toda la información que ustedes requieran y nos contactan y con mucho gusto. Carolina, muchas gracias por la organización del evento y pues Fernanda, de nuevo, muchas gracias y esperamos tenerte pronto por acá.
Gracias a ustedes.
Chao.
Jonathan, también muchas
Chao.
gracias. Entonces, yo creo que ya para la próxima, hasta luego.